DNA transcription has 3 stages, which are initiation, elongation, and termination. We begin transcription with the initiation stage. Here, transcription factors will recognize a promoter sequence and bind to the TATA box and form an initiation complex, which will attract RNA polymerase and initiate transcription. 35 base pair from the start line of transcription is the 35 box region. Once RNA polymerase binds to the promoter, it will unzip the double-stranded DNA. This will form a transcription bubble which consists with 2 strands, a coding strand and a template strand. The template strand is where RNA polymerase will slide over and produce a complementary strand to make RNA transcript. Once we enter the elongation strand, RNA polymerase will move along the template strand, which will read in a 3 to 5 direction, but will transcribe in a 5 to 3 direction. Transcription will have complementary bases to the template strand, but since it is not transcribing into RNA, the complementary will have uracil instead of thymine. As we now enter the termination stage, RNA polymerase will keep running down the template strand and keep making a complementary strand until it hits a termination sequence. This will stop transcription. RNA polymerase will release from the template strand and release pre-mRNA. Before transcription can happen, pre-mRNA must be processed and converted into mRNA. To convert into mRNA, we must have splicing, a 5' and a poly A tail. In splicing, this will remove introns from pre-mRNA. Introns must be removed because it will not translate into proteins, only exons will. In order for splicing to occur, a spliceosome has to form. Spliceosomes will bind to the ends of the exons, which will split the introns from the pre-mRNA and release into the nucleus to be degraded. To prevent degradation on mRNA, we need to add a 5' and a poly A tail. The 5' will be added to the 5' end, and the poly A tail will be added to the 3' end. The poly A tail consists of a group with a lot of adenine nucleotides. Now that we have removed exons and added a 5' and a poly A tail, we have mRNA that can now leave the nucleus and enter the cytoplasm for translation.